Hello everyone. This tutorial is for Bio 105 Exercise 8. This one is going to talk in a little bit more detail about genetic drift. So genetic drift is a change in the frequency of genes or of alleles in a population due to random occurrence. So in this case we have an example where we have two different colored beetles. Some of them randomly get swatted. Those beetles reproduce, and all of a sudden now, instead of having a 50-50 ratio of color in the beetles, we now have a 10-2 ratio of the red-colored beetles to the blue-colored beetles. This is not a selective pressure, right? The red beetles were not selected because they were better at surviving. They just happened to not get swatted. So, one of the first examples I want to talk about is something called genetic bottleneck. So it's basically when a population has a large reduction in its size. Okay, something kills off a large part of the population. Things like fires, floods, climate change, that sort of thing. It removes individuals from a population, and that reduces the occurrence of alleles. Okay, so we've got an example down here where you start out with a large population size. All right, we've got 5 different alleles in this population. Goes through that genetic bottleneck. We've greatly reduced the population. Now we've only got 2 different alleles in that population. And now, even though that population has recovered and gotten back up to its former size, the genetic diversity is much lower. So, a real-life example of a genetic bottleneck are cheetahs. They are African big cats. They have extremely low genetic diversity. They went through 2 genetic bottlenecks. Interestingly enough, they actually evolved in North America and then migrated through Asia into Africa. And during that time, those individuals that migrated were reproductively isolated and reduced in number. So they had a reduction of genetic diversity at that time. The founding population in North America subsequently went extinct. And then 10,000 years ago, at the end of the last ice age, they had another large reduction in population and went through another genetic bottleneck. So they have very low genetic diversity. Okay, our next example is founder effect. This can be caused by events that cut populations off from each other, and they often happen when a population moves into a new area and becomes reproductively isolated. So you've got individuals that move into a new area, perhaps an island, and they're no longer able to mix with the original founding population. And so what can happen is a gene that's rare in the original population can all of a sudden be much more common in the smaller new population. Vice versa can happen as well. A gene that's very common in the original population can become very uncommon in the new population. So our real life example here are Manx cats. They have shortened tails due to a gene that controls for the length of the spine. It basically tells it to stop making spine before it's done making the tail. So you have these little bobtails on these cats. The mutation that causes this is rare in the general population of house cats. But what you have is the Isle of Man off the coast of Scotland. That population of house cats was founded by a smaller population that had to be brought there by humans, and that smaller population had a higher incidence of this Manx gene in it. So what you have is the preponderance of cats on the Isle of Man are this Manx cat form. Now, you'll also see this kind of cat be relatively common because people have decided that it's cute and they will actually breed for it. But in this case, it was due to that founder effect. All right, so to wrap up real quick, genetic drift is a change in the frequency of genes in a population due to random occurrences. Genetic bottlenecks happen when a population is drastically reduced, reducing its genetic diversity, and the founder effect happens when a small subset of a population is reproductively isolated. I want to thank you all for listening today. We'll see you in lab.